TOB module. So today we will start with some terms or instruments used in the science of histology. Again, it's used also for the biology and histopathology. But as a start, we will discuss the terms and the instruments used precisely for histological studies. So regarding the first session of the histology will be the light microscope. So what we do study actually, we study either ميدان من بيت أحمد ورتيت يا مش نازم منينات هنا ويتي بس كيف كنا كان بنوصل بيت أحمد ده إفرن نوع. Regarding what we study, what's been لو مؤشرة أمان بيكشتبوا يصلاني online so I will speak to you about the items. We either study the structure in the organs or we study the function of them. So regarding the science related to the structure of the organs, we have the anatomy, which studies the forms and gross structure of the organs and tissues, and the histology, our science, which is related to the level, microscopical level of the studying of human body regarding the tissue and the cell microscopical structure. We have also the physiology, which studies the function of the body, how the different types of organs and tissues function in the body, and lastly we have the biochemistry regarding the chemical processes and substances inside the body. Regarding the introduction, we have the histology. Histology means web of tissue, and acacia is a web of tissues, and the logos means the study. So it is the study of the web of tissues inside the body. And how these tissues are arranged to constitute organs as aggregations of cells, forms tissues, and aggregations of tissues forms organs. Generally, tissues are made of cells and extracellular matrix. Cell can specialize to perform a particular function, and the extracellular matrix, which consists of many complex molecules, macromolecules found around the cells. Actually, these cells secrete or synthesize the extracellular matrix. Regarding the function of the extracellular matrix, first it provides mechanical support for the cells and fill the spaces between them. Second, it transports nutrients to the cell. It carries away catabolites and secretory products of the cells. Cells and extracellular matrix form a continuum. They are in always continuous interaction with each other. Regarding the tissue classification, you have to know in histology we have four main fundamental tissue parts. These are the epithelial tissue, connective tissue, muscular tissue, and nervous tissue. Amana general main tissue part in the body, and the rest of the body is composed of composition different percentage of these four main fundamental tissues. So most organs are formed by an orderly combination of these four types of tissues, except the central nervous system, which is only formed by the nervous tissue. Regarding the next slide, terms used in histology. What are the common terms used in histology that we have to be familiar with? So first we have specimen, biopsy, and tissue sections. Regarding the specimen, we have here in our studies human specimen, but the term is more general. So it could be animal, plant, or metal specimen. That we are considered with is only the human biological specimen, is any material derived from the human, such as blood, urine, tissues, organs, saliva, hair, and nail. while the biopsy is part of the specimen. يعني كواتا specimen is more wide and general term. The biopsy is specified for the medical studies. So this biopsy is a sample of cells, tissues, or organs taken from the body or by a surgeon, usually, ممكن a gynecologist usually, in order to be studied and examined under light microscope to determine the type of the tissues or the presence and extent of the disease. The third one is the tissue sections. So what is the meaning of tissue sections? Tissue samples, when taken from the body, or tissue biopsies, for example, they are too thick for the light to pass through them. So these biopsies have to be sectioned into thin sections in about 1 to 10 micrometer thickness. This allows the light beam from the light microscope to pass through them, or any type of microscope. It facilitates the passage of light beam through these tissues to be visualized and examined under the microscope. So we have also the relation between certain measurements that we use in our study. So you know that the meter is abbreviated as M, the letter M. Millimeter is about 10 raised to power minus 3 meter, while micrometer is 10 minus 6 raised to power minus 6 meter. The nanometer is 10 raised to power minus 9 meter. The angstrom is the smallest one, which is raised to 10.
The abbreviation of micrometer is wrong. The first M must be...
Yes. أخذنا كليرة. Well, it's not abbreviated. This in M. Yes. Okay. Regarding the types of biopsies, we mentioned that we have the biopsy, which is a sample of tissue or organ from the body. We have different types of biopsies. You have to know the terms used for the different types of tissue samples or biopsies. For example, we have smear. وش هي smear معناها؟ Smear بعربي بيوتري مسحة. You take a brush of a group of cells from the region of the cervix in the female genital tract, exactly from the cervix area, and this is sent for the histopathological study. For example, in cases of infections like human papillomavirus, or in cases of predisposition for malignancies, or the metaplasia, I mean the change of the tissues from the normal state to the precancerous state. And we have the curettage. Curettage is a term for a biopsy taken from the wall of the uterus. باشا عادة gynecologist ACA, it takes a sample by a metal instrument called curate. Our metal instrument that has a sharp end that will, when moved on the surface of the uterus, it takes part from the underlying endometrium. So this is also considered as biopsy. And we have needle biopsy. This type is taken by special needle or syringe. And this is taken from different organs. For example, the brain, the breast, the liver, kidney, and the tissue sample is again sent for the histopathological study. We have direct incision. For example, we take a part from the skin, a wound in the skin, or in the mouth, or the larynx. We have the endoscopic, in which we take biopsies through endoscopic instruments. Okay, when doing endoscopy by the surgeon or by the gynecologist. And we have transvascular. There are instruments that pass through the vascular system and have this special end that can take small biopsies from the wall of the vascular lining. So this is transvascular, sorry. Regarding the cells, you know we have different types of cells, many cells in the body, and they have different sizes. But in general, you have to know that most human cells are about 10 to 20 micrometer in diameter, about 5 times smaller than any object seen visibly by naked eye. And the sperm cell head is considered to be the smallest size cell in the body. It's about 4 micrometer in diameter, while the RBC, is about 5 micrometer in diameter. While the largest cell in the body is the ovum, which is about 100 micrometer in diameter. أما roughly بباسيكين جوكترين وجولترين زانيت. And the rest of the cells in between. But in general 10 or 20 micrometer. For example, in this slide you can see the size of the common cells of the body, while the oocyte is the larger size. Regarding the tissue preparation for examination, we discussed today in the practical session of the histology that before we examine tissues under microscope, we have to prepare them to be sectioned and stained in a group of tissue preparation steps. We will not mention the steps again here, just the highlights to remind you. But we have to say why do we prepare the tissues, basically what we prepare. So in order to study the tissue structure, we need to examine them under light microscope and the electron microscope, whatever. We have many types of microscopes, but we will mention here for the undergraduate studies the light microscope and the electron microscope. Tissues have to be prepared for microscopic examination. You cannot take a biopsy from the body and examine it directly under the light microscope or the electron microscope because of many reasons that we will mention. The steps of tissue preparation for examination, as the name, they are called tissue processing. TOB term tissue processing had to will be applied to the biopsies or part of the biopsies to be prepared for examination under light microscope. Why to prepare tissues? I think in the practical sessions we mentioned a few reasons, but I will just remind you. For example, the most common procedure in histological research is the preparation of tissue sections or slices that can be studied under light microscope. Under the light microscope, tissues are examined visually in a beam of transmitted light. For light microscope, we use a beam of photons, okay, for examination of the tissues. And because most of the tissues and organs are too thick to be examined, as I mentioned, under the light, and too thick for the light beam or the photons to pass through these sections to be visualized under light microscope, so they have to be sectioned and prepared before that, and apply them on the glass slides, histological slides, and examined finally under the light microscope. The ideal microscopic preparation is preserved, so this tissue has to be preserved from the degradation either by the enzymes present actually in the cell itself, the enzymes that are responsible for the degradation in a process called autolysis, or the enzymes in the bacteria, by the effect of the bacteria that causes putrefaction of the tissue. So we have to preserve the tissues from this process. The preparation of the tissue makes the tissues easy to be examined. However, during the preparation of the tissue, there is still problems like distortion, artifacts, changes in the tissue, and sometimes loss of components of the cell. It depends on how you prepare the tissues. يعني أنت شو استيقانك كثير شرطيها من أهم خاتية ideal bear. It depends on the physician and the technician who prepares the tissue. So it's not ideal all of the time. دائما artifact with distortion مش كتير أبيض. The steps, as we mentioned in the lab, in the practical session, start with the fixation, then dehydration and clearing, wax embedding, sectioning, staining, and mounting. And it's another terms used during histology, the plane of section. The plane of section, any biopsy or any type of tissues taken from the body, they have to be sectioned. But the sectioning procedure, which is done by the histopathologists usually, it have to be done in a certain plane, in different direction, that it will show the most details, the most ultrastructure in that tissue sample or in that organ. So we have different planes, and when you see the tissues under light microscope, you have just to examine them clearly and try to imagine in which direction the tissue has been, or the cell, for example, has been sectioned. So we have, for example, the longitudinal section. Let's say this is a tract in the body, for example, the urinary tract, ureter, or the respiratory tract, any tract, GI tract. So you can section it in this direction. So the section will look longitudinal, or you can cut it obliquely, or you can cut it transversely. So we have different planes of section. And when you examine, you have to remember which type of direction is this plane is sectioned. Example, when you have a three-dimensional structure, the three-dimensional structure also have different lines of section, so you can see the sections different from each other in each time. So you have to just imagine it. Regarding the instruments used in histology, the microscope, the light microscope used for the studies in the undergraduate students, for the medical colleges. The microscope, the light microscope I mean, is an instrument for viewing objects that are too small to be seen by our naked eyes. The definition of microscope means minute or very small, not visible to the eye unless aided by microscope. طبعا microscope يشتي من هو simple microscope, one set of lens يتيابوا, and we have the conventional compound microscope. كستل استشبك هاري when you say compound, that it has more than one set of lenses. We will discuss them. So we have the types of microscope. There are many types of microscope, ranging from the simple with the single lens, magnifying glasses, and the compound one, which is with high magnification power because it is more than one magnification set of magnifying lenses. Two basic types of microscopes are found. But we use here now mostly the compound light microscope and the electron microscope. The difference between these two techniques is that the resolution and the magnifying power is different between these two types of microscope. And magnification, you know, it depends on the set and the type of the lenses used in the instrument. While the term resolution, you have to know the resolution as a term. It means the simplest distance between two objects that you can differentiate the two objects from each other by naked eye. You can differentiate two objects, and this simple distance between them, this is called the resolution. أو تعريف العام as a general definition. So in our studies regarding the light microscope, it's found that the resolution power, the resolution power of the light microscope is about 0.2 micrometer. Basha. So it can see two objects as different objects, as separate objects in a distance of 0.2 micrometer. And it has a magnification power of 1,000 to 1,500. In some textbooks, and from 1,000 to 2,000 times it magnifies the ultrastructures in the cell. So this is a diagram or schematic illustration for the compound type of light microscope, which is used usually for the undergraduate studies. Laboratory can laboratory can you can see these are the different types, and these are the different parts of the microscope. We will mention them, and we mention the function of these parts. So regarding the first part is the eyepiece or the ocular part, which contains lenses for magnification, where you look through to see the image of your specimen. For example, this is the ocular part. This is one set of lens. It has three sets of lenses. This one, which is the eyepiece, and this is the four types of lenses, four different lenses I mean. These are called objective lenses with different powers of magnification. And these are sitting on a ring-like part, which is called the resolving nose. You can move it and you can put which type of magnifying lens you want exactly on the tissue specimen. This is the stage, here the stage, and it has a clips, certain clips. So you will put the glass slide on it, you will rest it there, and you put the clips so the glass slide will not move aside during the examination. And you have this part which is called head, sometimes it is called the body. Here this is the body of the microscope. This is the place that supports the instrument as whole, and you can hold it by hand and move it from one place to another place. And in Atwani is the most supportive part. You can hold it from here, the body, while this is the base, and this part is called the head, which lies near the eye lenses or the eyepiece. And we have here two knobs. One is called the coarse adjustment knob. This is the larger one, and this is the fine adjustment knob. The coarse adjustment knobs, when you move it, you can rotate it, so the stage moves ups and down. باشا وطبعا سير لأي سكوي you will examine the tissue or the slide. So if it is far from you, you can adjust the stage and make it clearer or nearer to your view. While the fine one, it will be used to focus the image. If you examine after the moving of the coarse, if you see the image to be blurred or not clear, you can use the fine adjustment knob, so it will be more focused. Image focus according to your eye, of course. And you have the third group of lenses here, which is called condenser lenses. It is directly beneath this stage. These condenser lenses, their main function is to condense the beam of light. You know, the base of the instrument contains the illuminator, the source of the light from the bead and a mirror. This light beam comes from here, and there is a mirror here, and so it will focus the light upward. And the condenser lens here, it will focus the beam of light toward the specimen, which is here on the stage. I'm a general description, but as parts. Eyepiece vascular, the arm, it supports the body tube and lenses, uses the arm to carry the microscope, you can hold it. And we have the body, which maintains the distance between the eyepiece and the objective lenses. Body لبيني eyepiece لبيني objective lens Coarse adjustment knob, as we mentioned, it moves the body tube or the stage up and down. The fine adjustment knob sharpens the image or focuses the image if it was blurring to your eye. We have the revolving nose piece, which contains the objective lenses. It is a rotating device with which we have 4 types of lenses. And we have the stage, of course, in which the glass slide is fitted to it. And we have the clips on the mechanical stage that holds the slide in place during the examination. Sub-stage condenser, which is the third type of the lenses beneath the stage, as we mentioned. And we have a diaphragm. Condenser. It is between the source of the light and the condenser lenses. It opens and closes on your demand. So the beam of light is so strong to your eye and it is affecting your eye, you can control this diaphragm. So you will maximize or minimize the beam of light that passes through the tissue and the object through your eye. And lastly, we have the illuminator, which is found at the base, as we mentioned. These are the general parts of the microscope, and you can later on return to your lecture and see the different parts. So your microscope has 3 magnification. I mean the compound one, the compound microscope. The scanning one, low and high. Each objective lens will have written the magnification on it. In addition to this, the ocular lens, the eyepiece has a magnification. So the total magnification, when you see the tissues, is the power of magnification of the eyepiece plus the objective lenses. This is the eyepiece. Magnification here is 10 times, 10x. It magnifies 10 times. While these are the objective lenses, scanning, 4x, low is 10x, and the high is 40. And we have another extra objective lens, which is used for oil immersion. We have the tissues. 1 drop of oil is put on the tissue, and later on you will examine it with the 4 type of objective lens, which is called oil immersion type. طبيعي شوار تايبا باشا objective lens ككليرا طبعا خد تحكمي PAKA جولاني توا يعني هو will start usually from the scanning type أو بربر زي KAK low and high power وردو زي KAK باشا ليرا أمانة هموي they are labeled باشا راني تابتي هو مسراوة باوري magnification كي لسه مسراوة so you can distinguish between them. This was regarding the magnification power. كأتوت مانشيا objective lens كسكان كتشوارا لقال ocular lens كتيانا وما أو ثابتة أو ocular lens it's always magnifying 10 times more so the result will be multiplied so the total magnification for example will be 40. For example in the case of high power we have 40 times multiplied by 10 is 400 times magnification power and for the oil immersion again is more. Regarding the electron microscope, in general, the difference between the light microscope and the electron microscope is that the resolution power of the light microscope, electron microscope is much higher than the light microscope, as we mentioned in the practical session. The rest totals, I guess they told you, and of course higher magnification power. For example, here it has a resolution power of 0.1 nanometer and a magnification power of 400,000 times. There is a huge difference between the capability of the light microscope and the electron microscope. In general, we have these two types: transmission electron microscope, which is the TEM, and the scanning electron microscope SEM. طبعا لحالة electron microscope because it is with high resolution and it shows the ultra structural details that cannot be shown on the light microscope, we need a special type of preparation. So the steps of tissue preparation will differ. These general steps are the main, but the types of reagents, solutions, fixative material, embedding material, all of them, they differ in the state of the light microscope to electron microscope. The main idea is that in electron microscope, you need very thin sections, very thin tissue sections to be examined under light microscope. And for these very thin sections, you need a different type of hardening or embedding material. For example, instead of the paraffin that you mentioned is used for embedding in the light microscope, here we will use plastic resin. Plastic resin هي مادة بوكو epoxy. لازم احتمال كان زيادة بسنة بوكو epoxy it is a very hard glue-like material. It hardens immediately. It will not need a temperature or it will not need a baking procedure. Increase in the temperature in order to be hardened, like in the state of the paraffin. Paraffin is a baking cake. While the oven in high temperature, later on it will be hardened. But the plastic resin, which is used in the cases for embedding, in the cases of electron microscope, you do not need the high temperature. This is the useful point. So you can keep many structures and many enzymes in your tissue sample the same as it and will not be distorted during examination of the tissue. Plus there's another difference, plus the steps of the tissue preparation. The instrument that is used, the microtome, for example, kind of practical session, microtome tandy. This is the instrument which is responsible for the sectioning of the tissue sections after preparation. Our microtome is different. The microtome in the case of the light microscope is with is steel blade, steel microscope studies. It is of knife or diamond. It is very sharp. Sometimes 40 to 90 nanometer is needed for the electron microscope. This is a very thin section. Each instrument unless you have a very good embedding material and a very professional steel, knife of diamond or glass, as in the microtome specialized for the electron microscope. The original form of the electron microscope فكرة كشفية لقوا جهاز as a microscope instead of light beam or photons they use electron beam. So the two types of the electron microscope instead of the light source they have electron gun. يعني بيسروا جهاز كبارتي بتاعه which is called the electron gun. باشا cathode بتاعه. The cathode is a filament of tungsten. Whatever any material metal material that can be heated to produce electron beam. عشان نحط جهازنا تنستل بكارين so it will emit this light of this beam of electrons. طبعا أو electron beam لأنه vacuum it will run with very high speed. باشا this vacuum has different voltage. So it will run with high speed and it will hit the tissue section. This is how the tissue sections will be seen. طبعا quality رسم مكان الالكترون مايكروسكوبات زوجو فرق هي quality رسم مكان لك لحالتك مو مسألة magnification. It's not just the matter of magnification and resolution. The colors is different. The colors in the electron microscope is usually white and dark, but still you can see the ultrastructures. أما واضحة. While in the light microscope, it is the usual H&E staining that is used in the basic routine staining of the glass tissue sections. طبعا وفي جهاز من جهازك رسمه كيتي. This is a transmission electron microscope. This is the electron gun. Area cathodic here and anode. Between the cathode and anode runs a high-speed beam of electrons. Do our projection at the tissue sample. Regarding the scanning, for the transmission scanning, transmission, the beam of electrons pass directly through the tissue sample and can transmit through it. Now tissue sample so some of the electrons are transmitted through the section. While in the case of the scanning electron microscope, this beam of electrons will not be transmitted into the tissues. It just hit the surface. All level away, the surface will be covered with a metal-like element or solvent, so it will just hit the surface of the tissue. It will just scan the outer surface of the tissue. In the next lectures and practical sessions, we will put to you in the slides what are the images of the light microscope and how it appears, and the transmission electron microscope and compared to the scanning. They are different. But in general, the electron is with higher magnification and resolution. Transmission زيادة البكارة هي scanning limited. بس scanning توجد بس أنتوي general شيء structure كبيرة. بقولهم أنا أجر سيلا كتير مثلاً specialized سيلا أتواني بس the body of the cell or the processes of the cell cannot see the content of the cell. لو حالتنا nucleus أبيني لا organelle أبيني. But if your study is just designed to know what are in the cell actually, what's happening, أو academic transmission. And this is the scanning electron microscope. هذا هو تقريبا فرق الكيوانية بس فرق الكلوايا يدي كان سيرفيس بشانية يدي كان ديتيل بشانية. And thank you. I'll put you photos of them. جماعتي في شركة ترمان هنا أجر الرسالة الثانية برون أتوان استراحة يورق لنا دقة شاركة وبقوم لكتشر ورقينا بس إيش رسالة هي أتواني بالتليفون كما عرفتي بس ما قلت من the tissues in the body are divided into 4 fundamental tissue parts. باشا أو جوايا they are joining in the form of organs with different percentage and preposition. So some organs, the epithelial tissue is more than the connective tissue, and other connective tissue is more. For example, in the nervous system is only of the nervous tissue, but then the nervous system is not composed of different percentages of the four main parts. It's only nervous tissue. So as we mentioned, the 4 different types are the epithelial tissue, connective, muscular, and nervous tissue. Rosmakani as a diagram or illustration looks like that, and in next labs, the images will be more clearer to you. These tissues, which all contain cells and molecules of the extracellular matrix, exist in association with one another and in a variable proportion and morphologies, forming the different organs of the body. For example, in the hand or in the arm, you can see different compositions the four main elements of tissues are found, for example, in the arm, hand, and forearm. مو هيك معين. Regarding the embryonic origin of the epithelial tissue, instead speaking for the first fundamental type, which is the epithelial tissue, you have to know the embryonic origin of this tissue. So the ectoderm gives rise to the epithelium of the external body surface, bone skin, type of epithelial tissue. This type is derived from the ectoderm. For example, other types of epithelium, like the epithelial tissue of the digestive and respiratory tract, part of them is derived from the endoderm. And for example, the mesoderm gives rise to the lining epithelium of the vascular system. Vascular system, the wall of the vessels, as you will study in the next modules. Again, it is lined by the type of epithelial tissue. Regarding the epithelium classification, boy epithelium benacito, boy exani epithelium tissue tron arrange, how it will be arranged and ordered in the different parts of the cell. You have to know a classification of this tissue. So the simple or the main classification is that the epithelium is divided according to the structure, and according to its function, organic or organic to two types. First, the covering epithelium, and second, the glandular epithelium. You can see that the covering one is the type of epithelium which covers the surface of the body, for example, in the skin, or line the cavities in the body, for example, the cavity of the GI tract, the cavity of the urinary tract, and any cavity in the body. While the glandular one is responsible, it is formed in the form of three-dimensional groups of cells that is responsible for secretion of certain enzymes and hormones. So they are acting as glands. Regarding the covering epithelium, we will start with the first type of the epithelium, which is the covering epithelium. In the covering epithelium, it is further classified according to number of the cell layers and the morphology of the cells in the surface layer. يعني ما to epithelial cell which have certain characteristics, as we will mention next. We can classify it according to how number of the cells are arranged above each other and the morphology of the cells, as in these slides. For example, the first classification is according to the number of cell layers. Regarding the covering epithelium, we have one classification according to the number of cell layers. يعني كم cell layer بس because the epithelial cells are usually a group of cells tightly attached to each other, next to each other, forming cellular sheets. مجموعة sheet So we have either one cell sheet or more than one cell sheet. Regarding the simple one, the one cell layer is called simple epithelium. To term simple epithelium based on معناه you have a single layer of epithelial cell, a single sheet of epithelial cell, which is resting on a basal lamina, and underlying it is the connective tissue. باشا أما هلا ناو كيف ما دام covering لا free surface either the free surface of the body like in the skin, or the free surfaces of the internal organs like the gastrointestinal tract or the urinary tract. So it is a single layer of cells, as we see here, or مجموعة cells. These are the epithelial cells, or basal lamina or basement membrane, and beneath it is the layer of connective tissue. While the compound epithelium, chelator compound, أجرهاته زيادة cell layer باشا. So it is more than one cell thickness. As you see here, this is the picture referred to it with this pinkish layer. You can see a group of cells here lying to each other, forming a cellular sheet. Another group of cells, another sheet, so sheet over sheet, so multiple sheets of epithelial cells. لمحة chelator compound epithelium, or another term, stratified epithelium. Basha. So this is the most important thing to recognize in the epithelial cells. Tonatwani H epithelial cell bizani chijoreka tanatani chanlera. This is the first point. According to the morphology of the cells, which is the second classification of covering epithelium, we classify the simple epithelium, simple epithelium, the epithelium which is of one layer, of one sheet, to 3 types according to the morphology of the cells, either squamous, cuboidal, or columnar. Squamous is something very flat, disc-like cells. They are very thin, attenuated cells. Those are thin, just like a paper. Okay, with a central nucleus which is bulging outside. The uterus is bulging outside of the cell. This is the squamous. While the other one is the cuboidal, so it is cube-like cell. It is square in shape with central spherical nucleus. And the columnar. columnar ما بس منشيها rectangular cell. Cell كبشوي مستطيلة rectangular. So these are the simple types. For example, this is the simple squamous one. You can see the orange layer here is the cellular sheet or the epithelial sheet. They are very thin, attenuated sheet attached adherently to each other, amasialic, and with a central bulging nucleus. While this yellow resembles the basal lamina or the basement membrane. Lera, cuboidal, simple cuboidal, the height of the cells is higher, but the width and the height of the cell is similar to each other, so it is cube-like with a central spherical nucleus. In the cuboidal, the nucleus will be situated in the base of the cell. So this is mostly rectangular, in which the height of the cell is more than the width of the cell, as in this slide. According to cell morphology, we have also again the stratified epithelium, compound Kash, or the stratified epithelium, which is more than one layer, again classified to the morphology, not only the simple one. But here we depend on the morphology on the top of the epithelium. مجموعة شيتها مجموعة ليرتها باشا. So ناجي تو بيصل layer كبيبين. You have to watch the surface epithelial layer. You can see the shape of the cells there and decide whether it is stratified squamous, cuboidal, or columnar. ليش نفكر شلونه؟ بطلع عملية إن شاء الله سلايد تيجي أبينا نواصل تربية فكرتها. But there is a very nice main classification. أما classification covering epithelium همويتي. ويتابع كان examito بشكل examito ده ورده لهمو لا لكوس لا كان أبينا so you will imagine the idea. We have epithelium, the covering one I mean, 2 types, simple and stratified, or simple or compound. The simple is either squamous, cuboidal, columnar, and we have one extra type which is called pseudo-stratified. أما الأخيرة باسيكين لنهاية المحاضرة كباسيكين. It is a private entity. شيء كبتفجيات. أصلاً لبعض text book نلقى compound ده نلقى simple ده. It is a different entity. But إيش باسيكين بو. But in some books it's set with the main simple epithelium. باشا أنجورانا طبعاً فقط كشيء ciliated or non-ciliated. كأني ciliatia, it is a certain microscopical feature of these epithelial cells. It is an accommodation of the cell. يعني feature كهني epithelial cell هاتي to do certain functions in certain types or in certain organs. لها مش هنا كانيا. So in certain organs, for example, we have simple columnar ciliated. يعني simple columnar ciliaci بسرعة. In other organs, we have simple columnar but non-ciliated according to the function of that organ. The pseudo-stratified is also mostly ciliated, بس بعد شوي شاهين non-ciliated. يعني cilia بس طبعا ciliacia, they are finger-like projection on the top of the epithelial cells. They are moving. They are responsible for the movement of particles of foreign bodies or certain substances from one part of the body to another part. باشا بجوغيتها. وتقريبا قوي هنا يعني حجم زوزو بشوكات 5 the height of the cilia. It's about 5 to 10 micrometer in length. باشا. Regarding this stratified is again has the same morphological classification. Here in the stratified, for example, the squamous has 2 types, non-striated and
كيراتين.
كيراتينايزد not striated ده ولا مصلحة هذول باسكين non keratinized and keratinized طبعا كأول keratinized ما بس ليه كيراتيني بس رواية تروج دون نوع وهي تفاصيل باسكين إن شاء الله في the next lecture yes pseudo stratified columnar and cuboidal her second بس most commonly columnar pseudo stratified cuboidal كمان لإحالة sedated هي وسببها باسكين ماشي لك okay regarding the feature of epithelial tissue general features أماني covering epithelium شيء common feature كاني closely packed polyhedral cells طبعا when we say polyhedral we mean multi-surface هتحط cell كتهب multi-surface هي بيوتر polyhedral بتلحق بالسيرفس هي بتشاري هي بالبنجي هي أو multi-surface طبعا ثابتيش يعني السيل كوبوصلة فقط مثلا هي شوار surface هي تين surface هي so it's polyhedral not fixed باشا with little extracellular material. Amount man offer main tissue shocanical which is the epithelial connective tissue, nervous and muscle. هميان as general common feature يعني aware pick هاتون from cell and extracellular material. Extracellular material خوي أوبران substance كسيلا كخوي بدور خوي دروسيا كا. It will synthesize it to protect itself. طبعا ما بس تيجي تيا هاي بس يمكن. Basha. So the difference between these four fundamental types of tissue is that the type of the cells and the amount of the extracellular material differs from one type to other. For example, in the epithelial cells, the cells are tightly adherent to each other in cellular sheets, so there is very little extracellular material in this type of epithelium. Basha of tissue. مثلا connective tissue we have abundant extracellular material, so it depends on the main type of the tissue. In the muscle we have moderate amount. In the nervous system there's none, very little extracellular material. يعني بعد textbook قال none أصلاً نيل. Okay, so it differs. Lera, it is little extracellular material. Epithelial cells are arranged in continuous sheets. We mentioned that. And we have another features in the epithelial cells that they have polarity. Again, the cell has polarity. Wasn't the biology is that the cell has two parts, apical part and basal part. For example, these are groups of epithelial cells, tightly adherent to each other with no extracellular space between them. They have a free surface here, which is the apical part of the cell. بشكل modification كان له يعمل whether microvilli, cilia, free surface and a basal part, which is sitting directly on the basal lamina. Basha, basal lamina شو sheet يبها أصلاً cell كبروسياكا. And beneath it is the other type of tissue, which is the connective tissue. Karata and feature polarity, apical surface, basal surface, and we have lateral surfaces. زيادة apical وبasal كان transporting material will move them to waste products بالاتجاه apical to basal, basal to apical. So this is cell polarity. Epithelium is avascular tissue. Avascular يعني blood supply near أو إذا قلت مجموعة sheet يورقي لepithelial cells. Whether single or stratified. أبيني blood supply يعني. The blood supply comes from the underlying connective tissue. The pure nutrition يبويت from the underlying connective tissue. عادة connective tissue is highly vascularized while the epithelium is avascular. عادة لو كنت تعرف شو كزور بطانية سواء as a blank, true false, باشا بويا underlying cloud. Epithelium adhere firmly to nearby connective tissue by a basement membrane, which will I discuss to you now. Epithelial cells have a high capacity of renewal. هذا التشو كان كزور بس renewal تيابيا وخصوصا the lining of the GI tract باشا كبردوام process renewal. The main functions of the epithelial tissue, it has a role in the protection, like in the skin, filtration, like the epithelium of the kidney or the renal tubules, lubrication, like the mesothelium inside the intraperitoneal cavity, secretion, like in the kidney, digestion, example the GI tract, absorption, excretion, transportation, many functions according to its site in the body, whether it is in the different organs, which organ it has the characteristic and the function of that specific organ. اگر بین با cell shape که cell shape که بگیریم ولی من یکم شد to recognize the type of the epithelium, the location of the epithelium, باشه بزنی تو QIK examination یکی under microscope, and you can decide whether this type of epithelium is simple or stratified again, depending on the shape of the nuclei of the cell itself. يعني أجر هذا cell كتسكويرمس مو flat there, so the function, for example, differs. Why it is flat? Because it is to facilitate diffusion. بوي لو شونانه diffusion man periostapy. باش لو شونانه أبيت. The cuboidal one, they are cube shape as we mentioned. They function in absorption. عادة أوجه السيلنا الabsorption عايشة secretion and absorption. باشا. Like example in the glands, the ducts of the glands, باشا, or the absorptive parts of the renal tubules. The columnar or the rectangular one, أمانة usually have cilia, باشا, for the, as we mentioned, for the movement or trapped material or foreign material, for example, in the respiratory tract, or the secretion and the absorption as in the lining of the intestine, which the GI tract is done by the columnar epithelium. What about the nuclei of epithelial cells? يعني ما هكو أمثلة أكو outline كمان ناسي. What about the nuclei? The shape of the nuclei is relevant to the shape of the cell itself. So the longitudinal axis of a nucleus is parallel to the longitudinal axis of the cell. Again, هاتو cell كت flattened بين nucleus كشت flat area. بس طبعا إما بس ما كل السيكونس وكمان توزع protrude كشكزور زور تاني كسيال كان سالفة لازمي كمان. Boy nucleus كتوزع برستندر كده. خوي still nucleus is flat. باشا. While in the cuboidal one is spherical. باشا. عادة شكل spherical area. And the columnar, it is elongated or ovate and situated at the base of the cell, centralnia. تو بشيوي nucleus كزور جار أتواني بشيوي cell كبزاني بول بالقوي إما وكو بس ما كل لايك microscope تواناي magnification or resolution man كمان. So most of the time in the electron microscope you cannot decide the outline of the cell. These cells are tightly adherent to each other and the walls have interdigitation between them. So you cannot decide whether this cell is columnar or cuboidal. You cannot see the outline. So return here in this state to the shape of the nucleus. أجر هاتو nucleus أكون centrabia, large, spherical nucleus, you can assume that this is a cuboidal epithelium. أجر هاتو cell أكون slide كتشون بين elongated, elliptical, ovoid, and located at the base of the cell, you can decide that this cell is columnar. جاري واشي أجر preparation slide أكون زور باش بيو technician أكون John Stein مشتي كردبه أو كاتا you can see the outline. باشا? So this is very important to depend on the number of the nuclei to decide first the shape of the cell, and it helps us also to decide the number of the layers. أجر هاتو تود أكيد نيجن layer تاع. When we stratified as an okay compound stratified, but you cannot decide whether how much layers have in this epithelium, which is important. وهي استفادية أكيد for diagnosis. باشا. لو حالتها أتواني county nucleus كانكي. You will see the nuclei bluish or purple in color. They are very clear. باشا you cannot miss them in light microscope. أو كانت أتواني county nucleus كانكي كي بو examine channel. Okay? So the nucleus is very important. Regarding the basal lamina as entity, she called basal lamina. Basal lamina is a layer, a very thin, very delicate layer that separates the epithelial tissue from the underlying connective tissue. Or different other tissue, شبيه connective tissue. بس عادة نجار epithelium connective tissue. And it is a line of demarcation first. It will distinguish the epithelium from the underlying connective tissue. It is a barrier. For the passage of materials easily between the connective tissue and to the epithelium. إلا selectively أو الخويبية رجعنا على مشكلة. Second, it is mainly formed by the cell itself, and epithelial cell like a whole excrete account. So it is a layer of extracellular matrix. Basha balanchita, collagen type, which is a characteristic type of collagens only found in the basal lamina. باش غير على السيل كاين وكو plain illustration اللي بيمين أما epithelial cell كده أم layer bacteria direct beneath it that supports the epithelial cell بس لا basal lamina. طبعا basal lamina is very thin. فعادة لهنا دناك زادن أو basal lamina not only like microscopic you cannot detect it. Why? Because it is very thin. تقريبا less than 60 نانومتر قبل بيتا ولا تكستبوك كان طبعا تزبط فيها الفيسبوك كان بس بحدود 40 to 60 نانومتر. It is very thin. باشا why not only detect it like microscope you can see it with electron microscope. ليرا ونمونا أجا see am slide okay this is an actual H&E slide يعني slide histology. You can see a group of cells adherent to each other. You can see the nuclei of the cells. باشا spherical nuclei احتمال الملزمة كانت تنجوات الواضح بالإرديانية. And these nuclei cannot distinguish the cell walls. Not only cell wall can be meaning, but شو spherical you know that this is a simple cuboidal epithelium. طبعا بجيريا أليرا beneath it. باشا beneath this layer is a basal lamina which is again cannot be seen because it is like microscope slide. باشا examined at the light microscope. What is the difference basal lamina and basement membrane? يواز يعتر لكلاسي بيولوجي وزامهر the basement membrane إكفران يعني كتيامي basal lamina كم يتبولاتا مدى تازل مثال medical college إكفران. So basal lamina, what is the difference from basement membrane? Basement membrane is thicker than the basal lamina. Basha boya basement membrane you can see it with light microscope. While basal lamina you cannot see it. Difference okay, aware just aware American thinner and one is thicker. عادة أجر do basal lamina join بالبيك هوا. أجر صدفة هاتو do epithelial type. Basal lamina epithelial tissue يعني sheet يعني. لقوا basal lamina another epithelial tissue هاتو join ولا بعد شوينا هي أو join بالبيك هوا without connective tissue between them. لو حالتها أستورتنا يعني. So the term will change from a basal lamina to a basement membrane. باشا بس كأولي definition كأوتا. لو حالتها إخواني بلاك مايكروسكوب بيبيني. لكن أخوي همان component يعني هل extracellular material وهل collagen type 40 ترى بلام double layer. Okay. Regarding the types of simple epithelium, we will count the classification of Akani Basake. And we will leave the compound epithelium for the next lecture, inshallah. Regarding the simple epithelium, as we mentioned, single layer of flat or squamous cell. أهم مجموعة cell كان يتشير they are flat and squamous, with central, somewhat protruding nucleus, with a basal lamina. Beneath it is the connective tissue. Location عادة, by the way, it's very important for the epithelial tissue to know at least 2 locations for each type of simple of epithelium. يعني هالجوريكي epithelium أبيت هو 2 locations ثاني دون منيلة ثاني أو مطلوبة للامتحان. Okay. So regarding the simple epithelium, the simple squamous I mean, it is found in the alveoli of the lung. باشا. It lines the free surfaces of the serous pericardium. شلو شيناها هذول pericardium. And it lines the pleura. Okay. And the peritoneum, lining the peritoneum is this simple squamous epithelium. Here it is called mesothelium. Now, can it was agoric? The simple squamous, the peritoneum are portrayed mesothelium. Lining of the blood vessels and lymphatics there portray endothelium. بعض الترم هي agoric according to the organ. يعني قبل بحقيقة لها مو organ كان simple squamous أخوي نقرأ أو شيء. But the terminology is to help you. The terminology is to help you to differentiate location from location. يعني من أجل endothelium شكل blood vessel هي أبي توريك بزاني من بس simple squamous epithelium أكيد lining بس لكانه كم باش أجل mesothelium you have to know this is the lining the simple squamous epithelium which lines the peritoneal and pleural cavities. Okay. Regarding their function, it's for transportation. I'm very thin, disc-like, so it is for diffusion, for exchange, gravity exchange. For diffusion or filtration. These are the cells of the lining of the alveoli, simple squamous epithelium. Alveolus, this is another alveolus. I'm sure it's very difficult to you to imagine it now, but later on لأن راستي المقولة الأخيرة إن شاء الله شكلها واضحة. And this is a lining of blood vessel. باشا, a lining of blood vessel which is lined by endothelial cells, the simple squamous epithelial cells, the flat cells that line the wall of the vessels. Endothelium. Again, and these slides are example of the mesothelium and endothelium. يعني أما capillary, you can see the blood RBCs here. This is a cross section. باشا, أما ليرة, just to imagine it. This is a longitudinal section of the capillary. Capillary كمان هنا كتمان كردوا laterally or sagittally. So this is a longitudinal section. You can see the wall of the capillary, the empty space, and the RBCs inside it. Masha? The walls are lined by the endothelial cells. These flat nuclei, Masha, these are the nuclei of the simple squamous epithelium. Regarding the simple cuboidal, وفرص كمان مشان نكشتر وكمان cuboidal cell functionality secretion and absorption عادة وش يعني secretion absorption تاعها لكواها and the cuboidal epithelium may be seen in the follicles of the thyroid gland. The thyroid gland هي the simple cuboidal epithelium and in many glands, ducts of the glands and on the surface of the ovary. أول يرسي location هي تتوخد وان هل بشيرة ولا بالي كموخد. Okay? Simple cuboidal, these are the thyroid follicles. This is a cross section through the parenchyma of the thyroid, so you can see these follicles. Structure they are lined by simple cuboidal epithelium. With central spherical nuclei. This is a cross section through urinary tubules. The urinary system, inshallah, in the next year you will study the urinary system. It has a group of tubules, like a cross section, and lining a cage here. Some of them are lined by simple cuboidal epithelium. You can see the central spherical nucleus and you can see part of the wall which is cuboidal. While this one here, tubular, it has simple squamous cell, flattened nucleus. This is the thin part of the loop of Henry. They are shorter and squamous in type. Going away to a cuboidal or squamous. Okay? The simple columnar type, cells of epithelium much taller compared to the width. Nuclei elongated, curved. عادة lining GI tracta. These are in the GI tract not ciliated, they have macrovilli instead. Macrovilli is apical surface, yeah? And some of them, like in the respiratory tract, they have cilia. These are rectangular cells, basal nuclei with cilia. So this is the ciliated type. So, for example, on the location of the non-ciliated, the specialized cells with macrovilli and sternal tracta, gastrointestinal tract. Some of them are found on the gallbladder. Their function is for secretion and absorption. While the ciliated simple columnar epithelium is like the upper respiratory tract, fallopian tube, the female genital tracta, and the uterus and central part of the spinal cord. Functional as mentioned here, secretory theater and the respiratory tract for theater functional with cilia for trap the foreign particles that enter the airways. And the uterine tube, fallopian tube, they are responsible for the movement of the fertilized ovum towards the uterus. And sometimes they have macrovilli as in the GI tract, which is for the purpose of absorption. The GI tract responsible for absorption. Lastly, we have the pseudostratified epithelium. كباسمان كروتمان ده unreliable simple بس هو جار يعني single entity بالتالي ده specific type of epithelium. It is not a true stratified, يعني not stratified or simple. كسيري أكيد شو كل stratified شيء. Looks like stratified. You will see the nuclei at different levels, but actually they are themselves, all of them are attached to a single basal lamina all of them, they are with the same height, attached to single basal lamina, single layer of cells, but they nuclei at different levels. So it gives the impression كسيريا كيوا أزاني certified. كتوزي nucleus كان لأمام cellular sheet near لأمام levelania personisma. So you assume that it is stratified, but it is not stratified. Well, actually, حتى بسمبل إيش اللي بعد textbook هذاينا. So here you can see, for example, this is one nucleus, this is another nucleus, this is another nucleus. The cells, all of them are attached to the basal lamina, but not all of the cells reach the apical surface. حميانا كان apical surface. باشا I'm George. عادة ciliated cilia if you see this section باشا بس السيولة كانوا أمانة surface has ciliated accommodation. عادة الكيو start is found in the respiratory tract. The pseudostratified ciliated is found in the respiratory tract. Boku diagram our area, boku actual slide our area, and these large cells, these are the mucus secreting cells. عادة the respiratory tract or the GI tract or cellular epithelial goblet cell. Function here, mucus which is a viscous material, helps to lubricate the surface or to wash the cilia. And the function, according to the organ, respiratory, function. Well, regarding the non-ciliated pseudostratified, it's found in certain areas, like the auditory tube. That is difference in male and in the male urethra. Lemjora, it is pseudostratified but without cilia. This is a high power slide for the pseudostratified with different level nuclei. And thank you. Again,